Hello, everyone, and welcome to ACS Webinars, connecting you with the best and brightest minds in chemistry, live every Thursday from Washington, D.C. I'm Michael David, and I'll be your host for today's broadcast, which is being co-produced with Biochemistry, a journal from ACS Publications. During the next hour, we will be learning about an area in molecular engineering that holds great promise. Our presenter is Rohit Pappu, who is the Edward H. Murdy Professor of Engineering and the Director for the Center of Biological Systems Engineering at Washington University in St. Louis. He will be discussing the physical foundations of phase transitions, especially that of intrinsically disordered and ordered proteins. Now, our moderator today is Sarah Smaga, who is the coordinating editor at Biochemistry. Sarah received her PhD in molecular biophysics and biochemistry from Yale University in 2018, and her BS from the Lyman Briggs College at Michigan State University. She also serves as the project coordinator for the Center for Genetically Encoded Materials, an NSF-funded center for chemical innovation. And now I would like to turn it over to you, Sarah, to get our presentation started.
Thanks, Mike. Hi, everyone, and thank you for logging in. I want to extend a warm welcome on behalf of all of us at Biochemistry. We're excited to co-host this webinar because phase separation is tremendously exciting from a biochemical perspective. Although cellular features like Cajal bodies have been observed under the microscope for over a century, and it's known that these droplet organelles are important biologically, we're only just starting to understand how these structures form and how they function on a molecular level. Today's talk will delve into the biophysical basis of phase separation, and I'm excited to introduce our featured speaker, Rohit Pappu. Dr. Pappu is joining us from Washington University in St. Louis, where he is the Edwin H. Murdy Professor of Engineering and Director of the Center for Science and Engineering of Living Systems. Recently, Dr. Pappu was named Fellow of the Biophysical Society for ingeniously implementing polymer physics approaches and molecular simulations to characterize intrinsically disordered proteins, which I believe we'll learn more about in this presentation. And with that, I'll turn it over to you, Dr. Pappu.
Thank you, Sarah. I will jump right in. The topic of today's webinar is phase separation of multivalent proteins. And as Sarah noted, the motivation for this comes from the observation, almost a century old now, that there are numerous organelles that do not have a vesting membrane and hence are called membraneless organelles that facilitate the spatial and temporal organization of cellular matter. And this particular schematic shows you a few examples drawn as blobs of the different types of bodies that over the years have come to be known as membraneless organelles. These days, membraneless organelles are also known as biomolecular condensates because, quite simply, they are thought to concentrate specific types of biomolecules via processes that very much resemble the condensation of liquids. And this is a term coined by Mike Rosen and Tony Hyman. An example of a biomolecular condensate would be a nucleolus where ribosomal subunits are actually assembled. So the sites for ribosomal biogenesis are the nucleoli. They are gigantic organelles inside the nuclei. And these would be examples of what Cliff Brangman would refer to as multiphasic coexisting liquid-like condensates. The term liquid sort of comes from observations now that are exactly a decade old, made when Cliff Brangman was a postdoc in the lab of Tony Hyman, demonstrating that P granules, which are organelles that control the asymmetric cell division in worms, basically show the properties of concentrating the key biomolecules into these puncta that are shown as bright foci, but also showing the feature of dripping and flowing and fusing that is characteristic of liquids, and also the ability of being rounded up. Now, this was 10 years ago, and I think at that time, the emphasis was really on the liquid-like behavior of these condensates, but 10 years since, in a rapidly moving field, I think we've come to appreciate that the liquid-like features are but one aspect of these different types of condensates. There's an assortment of material properties that one can envisage for these condensates. And what this leads us to is the obvious question, which is how do we think about the driving forces for condensate formation? And so This talk will build on the observation that at least in stylized systems, it would appear that facsimiles of biomolecular condensates form via what is referred to as phase separation of multivalent protein and RNA molecules. Now, this might seem like a mouthful, so what I'll try to do is sort of walk us through the particular physics that underlies phase separation and what we mean by multivalence. So here is a cartoon that basically depicts the essence of what phase separation is all about. So shown here on the left are polymers sketched as these sort of noodly looking structures in blue that are part of a milieu, could very well be the cytoplasm or the nucleoplasm. And in the presence of some trigger, and or an overexpression of the protein or polymers in question, shown in blue, effectively one sees the formation of a droplet that basically is a condensate or a membraneless organelle that basically concentrates the molecules of interest here, leading to the coexistence of a polymer/protein rich phase that coexists with a polymer deficient phase, which of course will now be solvent rich. So this is the sort of basic phenomenon. There are numerous analogies that people will come up with. You know, you go to a bad Italian restaurant, they basically will insist on having vinegar floating in olive oil. So there's an example of demixing. So these are examples that we relate to as exemplars of phase separation. What this talk will try to focus on is a unifying framework that is not fixated on the purported structural features of the proteins or RNA molecules in question, but instead focuses on the fact that there should be a unifying framework that's good for describing the phase separation or phase transitions of the types of molecules that drive condensate formation in cells, which generally have the feature of being multivalent. And by that, what we mean is a high repeat number, if you will, of sticky motifs or domains that provide specific interactions that we have come to know and appreciate in biology. To bring a physical framework to bear on these on this problem, we have borrowed quite heavily from the field of polymer physics, and in particular from the fairly modern, comparatively speaking, field of associative polymers, which generally have an architecture that one can describe as being a stickers and spacers architecture. And the thinking here is quite simple. Shown in blue are the so-called stickers. These could very well be short linear interaction motifs that are very well conserved in the context of intrinsically disordered regions. They could be well-folded domains, which we see quite a bit of. Examples would be SH3 domains, SUMO domains, PDZs, and the like. And these stickers that provide specific interactions will then be interspersed by spacers that could, again, be in either intrinsically disordered, or they could be other structural domains that provide a sort of scaffold. But the key point here is the number of these blue stickers, and as the number increases, so does the valence of the sticky regions or sticky motifs. The key defining feature of associative polymers is that stickers are referred to as being such because they preferentially interact with one another. And so there is a bit of a sort of Houdini act here, which is that the key component that actually determines these interactions, which is the solvent, is not shown in this schematic. But the way to think about preferential interactions is the stickers have a choice of interacting with themselves. They can interact with the spacers, or they can interact with the solvent. And in this three-way interplay, a motif or a domain emerges as being a sticker because it preferentially interacts with itself. And these interactions generally show up in the form of physical crosslinks that also then confer upon the higher order structures a network-like architecture, which we will come to later during the talk. So the first order of business then would be, given a sequence, how would we tell stickers apart from spacers, and what would be the criteria by which we would make such judgments? I will sort of go to a fairly challenging system to address this issue first, because in systems such as the one shown in the bottom, where there are well-defined RNA recognition modules or folded domains, identifying the stickers is generally a bit more easy because the well-folded surfaces of these domains generally have what we think of as being hotspots for protein-protein or protein interaction, RNA interactions. And so identifying stickers would seem at first glance to be a relatively easy adventure, if you will, when confronted with folded domains. But of course, many of these proteins tend to have these so-called low complexity domains and or intrinsically disordered regions appended to the folded domains. This has led to the somewhat misleading view that intrinsically disordered regions are the primary drivers and sole drivers of phase transitions, and that is hopefully a myth that will slowly get sort of fallen by the wayside. But for now, what we'll do is we'll focus on these LCDs or low complexity domains and ask the following question. Given a sequence such as this, which is taken from the low complexity domain of HNRNPA1, a stress granule protein, you will notice the sort of parsimonious usage of amino acids. A very small number of amino acids are being used here, and that is why these are called low complexity domains. These typically tend to have the defining characteristic of conformational heterogeneity, meaning that in some global sense, there might be sort of a defined ensemble of conformations, but generally in solution, these types of molecules fluctuate between an assortment of different conformations. And so then the question that we're challenged with trying to answer is, how do we identify stickers in these low complexity domains? In order to do so, we will actually sort of draw upon the thermodynamic principles of phase separation, which plunges us into thinking about a coexistence curve, which basically defines the phase boundary between a dense and a dilute phase. And what I will do is take a simple example of what I'll call a pseudo-binary mixture. And the reason for the term pseudo is that, of course, the solvent is not just water. It has a lot of other components that actually turn out to exert quite an influence on the phase behavior of mixtures. But we'll sort of pretend, as good theoreticians will, that we can simplify the system into being a protein plus solvent mixture and therefore a binary mixture. So a typical coexistence curve would look something like this, shown in green. And basically, this would be plotted in a two-parameter space where the x-axis here refers to protein concentration, and the y-axis now refers to some measure of interaction strength, the idea being that the interaction strengths between molecules is weakening as we go up, and therefore this is referred to as the inverse interaction strength. Now let's do the following thought experiment. We'll pick a protein concentration that places our system, our protein plus solvent system, somewhere in the two-phase regime as marked by this asterisk. And the key point that emerges as a consequence of phase separation is that the one-phase mixture is unstable. So within this two-phase regime, what we will observe is that the protein separates into two phases. The blue phase would be the so-called dilute phase, wherein the protein's essentially sort of behaving like a one-phase system dissolved in the solvent, and shown in magenta would be the circle that represents the dense phase, where now we've concentrated proteins into a coexisting dense phase. There is a line that we can join that connects these two coexisting points that will be referred to as the tie line. That essentially is the line that connects the points of equal chemical potentials, because the key point to remember here is that the blue and the pink circles are not some random points drawn on some boundary. They happen to be the two phases where the partial molar free energies of the protein are equal to one another, and that partial molar free energy is just a verbose way of saying chemical potential. Now let's do the following experiment, which is to go back and sort of ask, what are the concentrations within of the protein in the dilute and the dense phase, and we'll refer to these as these values as Csat for a saturation concentration and C dense for the concentration of the protein in the dense phase. So if we were to change our solution conditions now, or our protein concentration rather, where instead of being at the red asterisk, we move over to the orange asterisk, the protein still undergoes phase separation, and what we reproduce for concentrations in the dilute phase will be the saturation concentration Csat, and what we will reproduce in the dense phase will be the value C dense. The droplets will simply grow in size, but and the number of droplets will also increase, but the concentrations within the droplet will stay the same as C dense, and similarly the concentration in the dilute phase will remain as Csat, and this will be true as we sort of continue on and approach the phase boundary. Now, should we change the interaction strength, so now we have a sort of new set of solution conditions, if you will. Now we have different values for Csat and C dense. So in essentially the saturation concentration clearly changes as we change our interaction strength. So in a binary mixture, the defining attributes of a system that undergoes phase separation is the presence of this concentration threshold we refer to as Csat, or the saturation concentration, which is the concentration of the protein in the dilute phase that coexists with the protein in the dense phase. And so that saturation concentration is a threshold concentration, stays fixed as we titrate protein concentration within the two-phase regime. Now you can do this type of a measurement for a series of interaction strengths, which, for example, you can modulate by changing temperature or changing salt or pH, etc., and you get this sort of full coexistence curve. Okay. With that background in hand, what we end up with is a sort of zeroth order defining criterion for delineating stickers versus spacers, and that would be the requirement that in an intrinsically disordered region, if we were to make changes to stickers, because they provide the specific interactions that drive phase separation, changes to stickers, whether in terms of their identity or in terms of their number, will change the saturation concentration. Conversely, if we were to make changes to spacers, then what we will impact are really sort of how the stickers are brought together, and therefore the material properties of the condensates and also the overall cooperativity of the phase separation. So with that in mind, let's sort of jump ahead and think about a few archetypal intrinsically disordered regions. So here I show you two examples, the protein FUS and the protein EWSR1. These are proteins that have two distinctive domains, a so-called prion-like domain shown in blue, an RNA binding domain that encompasses intrinsically disordered regions shown in gray, and of course folded RNA recognition modules. The sequence has been bar-coded, if you will, with tyrosines and arginines for a reason that will become clear in the next minute or so. The work that I'm going to show you is the result of a collaboration with the Hyman and the Alberti labs performed by Tia Wong in a postdoc in the Hyman lab. The theory was driven by Jungmo Che, a former postdoc in my lab, and a lot of the computational analysis performed by Alex Holhouse, currently a postdoc in the lab. So let's do an in vitro experiment where basically one titrates the concentration of protein in the test tube. These are microscopy images, and basically what one observes is the presence of an apparent saturation concentration that's around 5 micromolar, one can then continue to sort of probe the validity of this being a saturation concentration threshold by way of scattering experiments or cuvette-based experiments that essentially show the onset of turbidity around 4 to 5 micromolar. So there is a saturation concentration. The key point is that this can be measured not just for FOS, but at similar solution conditions, so 75 millimolar potassium chloride, for an assortment of proteins that belong to roughly the same family. And what one observes is that the measured saturation concentrations shown here in blue clearly depend on sequence. Okay, so much of this protein is essentially intrinsically disordered. So, and a very interesting question that arises is, what are the sequence features which from some 30,000 feet look fairly similar to one another, but what are the distinguishing features in terms of the sequences of these different proteins that give rise to essentially a 2 order of magnitude variation in the measured saturation concentrations? Now, just as a sort of tantalizing fact, we want to make the point that, you know, the measured in vivo concentrations of these proteins also seem to show a trend line whereby you make more of the proteins for which there is a higher saturation concentration. Nothing more should be read into the depiction of these orange bars. So the question we'll try to answer very quickly is, what are the physical determinants of the sequence-specific values for the saturation concentrations? So some bioinformatics analysis helped point us in the right direction, because what Alex Holhaus and Jungmo Che did was to analyze the compositional biases in a variety of different proteins in the human proteome, and observed a very interesting feature, which is that in general, in the IDR-ome, if you will, the combination of arginines and tyrosines tends to actually be quite small, as depicted by the fact that most of the protein sequences actually live in the sort of far left corner of this plot. But what's striking about this FOS family of proteins, or FET family of proteins, depending on your preference, is that they clearly have a higher number of tyrosines and arginines. And I should clearly point out here that we're tabulating raw numbers. We're not normalizing this by sequence length, because that's actually an important part of defining the valence of stickers. So this led to us asking the following question in collaboration with the Hyman and Alberti labs, which was to ask, well, okay, since tyrosines and arginines seem to be quite prominent in this, let's start making some fairly blunt-edged changes. So basically what one observes is that if you, at the concentrations where one measures a saturation concentration for the full-length protein of 2 micromolar, if one were to perform measurements of the PLD alone, which has very few arginines, one doesn't detect any phase separation. Conversely, if we do the same measurement with the RBD, or the RNA binding domain alone, again, one does not detect phase separation. But if one were to mix these two domains in trans, you calculate or you measure out a joint saturation concentration that's 15 micromolar, clearly indicative of the fact that the complementary interactions between whatever the components of the PLD and the RBD are required to drive phase separation. And of course, putting them together in a single chain appears to enable the cooperativity of phase separation, as evidenced by the significant lowering of the saturation concentration. So that led us to postulate that the stickers in this particular case were actually the tyrosines and arginines, and here is simply reorienting you toward the chemical structures of tyrosine and arginines. And one of the first things that you will look at by dialing into the orange boxes that sort of highlight the functional groups is that you will say, oh, wait a minute, the tyrosines are these pi systems. The arginine, of course, has this delocalized cationic moiety, and therefore it is a cation, and therefore what must be driving this are cation-pi interactions. So what Jungmo said was he stepped back through the theory and said, well, let's not get beholden to calling these things anything. We'll simply stipulate that the stickers in the PLD are the tyrosines, the stickers in the RBDs are the arginines. And so if we now actually do a theoretical analysis based on the theory of associative polymers, where we try to predict the saturation concentration strictly based on the numbers of stickers, we actually, where we also impose a constraint, we don't have to, but in this particular calculation we did, that the stickers, the interactions that are driving phase separation are purely the heterotypic interactions between the stickers, namely the tyrosines and the arginines. The prediction that emerges is that the saturation concentration should simply go as the inverse of the product of the number of arginines and tyrosines. It's an elegant theory. You essentially write down a free energy for the system that's undergoing phase separation, but the upshot is actually very, very simple. And if you now go back and quantify the measured saturation, the correlation between the measured saturation concentrations and this parameter that basically is quantifying the impact of the valence of the stickers, you see a pretty remarkable linear correlation indicative of the fact that to 0th order, the determinant of the driving forces for phase separation in these FUS-like proteins is really just the number of tyrosines and the number of arginines. So this led us to sort of asking the question of why tyrosines, why arginines. There are several ways to skin this why cat, if you will. And so we sort of said, well, we can ascribe intrinsic multipole moments to each of these stickers. These stickers can have a charge and therefore a multipole moment, a monopole moment. They can have a dipole moment, they can have quadrupole moment, which essentially sort of talks about the in-plane arrangements of dipoles with respect to one another. And the reason this becomes important is that one can actually start to write out a sort of hierarchy of interactions whereby charge-charge interactions go typically as 1 over r, charge dipole interactions go as 1 over r square, and so on. And so the hypothesis then becomes that having stickers that can encode this hierarchy of interactions now enables to sort of draw the molecules at long range and then also provide the specific cohesive interactions that lead to defining crosslinks between stickers by way of short range interactions. So this also led to sort of some interesting thought experiments, which is that one can compare measured multipole moments of, let's say, tyrosine to phenylalanine, and one of the things that jumps out, of course, is that the in-plane hydroxyl gives the tyrosine a substantial dipole moment, whereas the phenylalanine has a negligible dipole moment. Similarly, electronic structure of the guanido group gives the planar guanido moiety a substantial quadrupole moment, whereas the amines, the other cations, namely those from lysine, will have essentially just a monopole moment, maybe a negligible dipole moment, but certainly no quadrupole moment of significance. And these are all very rough numbers and just sort of culling data from sort of standard measurements of these quantities. But the prediction that would emerge is that if we were to substitute arginines with lysines or tyrosines with phenylalanine, we must weaken the driving forces for phase separation. And that's a prediction that's directly testable. So here again, to reorient you, is the saturation concentration measured for wild type FUS in 75 millimolar potassium chloride. One then substitutes all of the tyrosines in the PLD to phenylalanine, and you can see an uptick in the saturation concentration, clearly indicative of the fact that phenylalanines make for weaker stickers when compared to tyrosines. One can do the equivalent sort of substitution whereby the arginines, which have the higher quadrupole moments, are substituted with lysines. And again, you see that there is an upshift in the saturation concentration, of course, and if you make the sort of double sets of substitutions, there's actually a pronounced increase in the saturation concentration. And again, a reminder, the driving forces for phase separation weaken if you measure an increasing saturation concentration. So basically, one theme that emerges is that without the consideration of specific structural features of these molecules, one can develop a zeroth order understanding of the driving forces for phase separation by systematic experimentation and theoretical work that in this particular case identifies the cationic and aromatic stickers as encoding a hierarchy of interaction ranges and strengths. And this is a finding that's actually completely synergistic with work, beautiful work that has come out of the Julie Foreman K lab. And these three papers here sort of are useful reads for those who are interested. But I do want to sort of make sure that I'm not conveying an erroneous message that we should abstract FUS family proteins as sort of diblock polymers of tyrosines and arginines, because in fact, the intervening residues of, namely the spacers, are actually important determinants of material properties and actually the cooperativity of phase transitions. And so if I can remind you, our criterion for adjudicating on spacers would be that they don't have a material impact on saturation concentrations, but they should have a material impact on material properties. As a way of sort of testing this, what Zia did was, based on sort of various prescriptions we came up with, substituted glycines with alanines, substituted serines with alanines, and substituted glutamines with glycines. Now, these are all, if they are stickers, they are sort of auxiliary stickers. And the measured saturation concentrations for these basically stay wild type like. So there's no impact per se for these changes on the saturation concentrations, but it turns out that they do impact the material properties as measured using a fairly crude approach, namely fluorescence recovery after photobleaching, which really quantifies not the various moduli that you would want to quantify for these condensates, but basically it helps quantify the rates of exchange of molecules across phase boundaries. And what you find is that the recovery is fairly rapid in these condensates when you first prepared them, but they do undergo a hardening transition because essentially there is a significant amount of cross-linking that's happening inside of these condensates. And so the recovery after photobleaching sort of plummets down to absolutely nothing if you let these condensates sit for a certain amount of time. But that feature is actually altered materially if one changes the serines to alanines. And in fact, if the glutamines are substituted with glycines, now you actually get what we sort of humorously refer to as immortal droplets, in the sense that, you know, the recovery rates don't show any kind of time dependence or aging dependence. So the key point is that the rates of these molecular exchanges are tied to, of course, the rates at which physical crosslinks are made and broken. And so the more crosslinks you make, sometimes it becomes harder to actually break them. And so one starts to see aging behavior, which we can discuss in the Q&A if people are interested. So intrinsically disordered regions, I think we sort of have a pipeline to help identify stickers and spacers. There are going to be examples, more easy to understand examples of, you know, associative polymers that have folded domains connected by intrinsically disordered linkers. I'm showing here the classic example that came from the work of Mike Rosen, where poly-SH3 domains bind to poly-PRMs, where PRMs are proline-rich modules, and then one sees the appearance of spherical condensates. But the key point is that while that work focused beautifully on the effect of valence, namely the number of SH3 domains and the number of PRMs, it didn't necessarily shed light on the impact of the linkers. And in collaboration with Mike, we demonstrated that the properties of intrinsically disordered regions, which is quantified in terms of what we refer to as effective solvation volumes, which is, you know, how favorably do these linkers interact with the surrounding solvent or not. That effective solvation volume, which becomes large and positive if these linkers really want to interact with the solvent, directly impacts the types of condensates that are formed. So the linkers are agnostic about interacting with the solvent. One sees what happens on the left, which is for the same valence and same interaction strengths, the formation of spherical condensates in simulations. Whereas if the linkers want to preferentially interact with the surrounding solvent, one can make this highly connected network that would essentially resemble what many people would define as a gel, basically doesn't undergo a condensate formation or phase separation, whereby you see spherical droplets forming. So basically what this says is that stickers and spacers come in different flavors, and that there's possibly this evolved synergy between between structured domains and disorder in terms of defining the identities of stickers and spacers. I want to sort of end with two quick themes. One is that biomolecular condensates actually have hundreds of distinct protein and RNA components. And it's very easy to get caught up in sort of the nitty-gritty of, reconstituting the phase behavior of a single system, sort of a binary system. But it's worth remembering that the hull bodies that Sarah mentioned, or the nucleoli that I mentioned earlier, these are molecules or components that actually have very large numbers of distinct protein and RNA molecules, and so these will have, roughly hundreds to, in some cases, thousands of distinct molecular components. So in order to start approaching the sort of biologically relevant question of how we think about multi-component systems, we've developed a lattice engine, and we here being Jungmo Che and Furkan Dar. Furkan is a graduate student in the lab. This is a way of coarse graining representations of biomolecules and doing large-scale simulations of these molecules on lattices. For those of you who are interested in playing with this and trying to adapt your particular molecular system to these coarse-grained architectures, the code is actually available on GitHub for free. Feel free to contact us with any questions you have about usage. But what I will do here is actually show you a very important generalization of the concept of saturation concentrations that we'll have to think about going forward with regard to multi-component systems. And what I'll do is take a very stylized system, namely nucleophosmin 1, which basically forms the outer layer of nucleoli, perhaps one of the more important scaffolding proteins in the nucleolus, that interacts with various arginine-rich peptides. In this particular case, we'll focus on a peptide called RPL5. And the Kriwaki lab in St. Jude, in a series of beautiful papers, has shown that one can reconstitute facsimiles of the outer layer, the so-called granular component of the nucleolus, using a truncated version of nucleophosmin referred to as N130. Shown here in red are these acidic tracts that make up the N130 molecule. It's a pentameric molecule. It's got folded domains that essentially sort of serve as this hairy colloid, if you will, that display these intrinsically disordered regions, which then interact with arginine-rich peptides to form condensates in vitro. Okay, so here would be our lossy instantiation of the underlying architecture. These shown here are the acidic tracts and the types of specific interactions that they can make with the arginine moieties on the RPL5. And what emerges is actually a very interesting feature for the phase diagram. Gone is the fairly simple sort of inverted parabolic structure, but what I'm showing you here is what the phase boundary will look like in the plane where we titrate RPL5 concentration. We use curious units like inverse voxels because we are doing simulations on lattices. And then in the y-axis, we have the concentration of the N130 proteins. And what emerges is this closed loop architecture for the phase boundary. So the low concentration arm would be where my cursor is sort of floating around here, and the high concentration would be where it's floating about here. And then there's also shown in green is what we call a percolation line, which basically defines the system forming a droplet spanning network. So what this says is some very interesting sort of features, which is that the closed loop will allow for re-entrant phase behavior, whereby one can fix the concentration of RPL5, go through the two-phase regime and sort of come out on the other, or fix the concentration, yes, of RPL5. And as we increase the concentration of N130, we can sort of come out of the two-phase regime. So the first message then would be that this green line, which defines percolation or the formation of a system spanning network, actually refers to the formation of what we refer to as network fluids. That means these molecules are actually connected to one another in a network in the fluid. They're not randomly organized around one another. The second point is that we now have phase separation that is driven mainly by heterotypic interactions, and in which case our phase diagrams will actually have a very interesting and curious property, and I'll sort of end by walking you through these features on these ellipses. So here is an elliptical phase diagram. We'll say we have two molecular components, A and B. Each of these lines corresponds to fixing the concentration of B while titrating the concentration of A. And we sort of go through the two-phase regime. The red dots are the dilute phase, the yellow dots are the dense phase, and these orange lines are the so-called tie lines that connect these coexisting phases. Now let's do the measurement where we measure the concentration in the dilute phase that coexists with the condensate. Where in the binary mixture we would actually have gotten a thresholding behavior, here we actually start to see an increase in the concentration in the dilute phase with the bulk concentration that reflects the fact that heterotypic interactions are actually dominating this particular system. We can change the slopes of the tie lines, and we can actually get some very curious behavior in terms of the concentration of the dilute phase vis-a-vis the concentration of the protein that we're actually titrating. So if one were to do in-cell experiments expecting to see saturation concentration thresholds, if the organelle in question, or the condensate in question, is driven primarily by heterotypic interactions, one is in for rather an unpleasant surprise, as shown beautifully by Josh Reback and Cliff Brangwyn in a preprint that's sitting on bioRxiv right now. But there is a limit where, let's say the tie lines are actually vertical, where now basically what happens is that B is mainly a ligand. It's not sort of it's basically preferentially interacting with these A's when they're in the dilute phase. Then one actually recovers the standard expectation of a saturation concentration, with the difference being that depending on the concentration of B, the saturation concentration threshold will shift up or down by a phenomenon known as polyphasic linkage. So what this demonstrates is that phase behavior in cells is going to be quite a bit more complicated, and it will be sort of governed by a combination of stoichiometry, by affinities, by valence, and also by the structures within condensates, which will be defined by the extent of cross-linking within condensates. It turns out that the stickers and spacers framework can also be applied to thinking about the interaction of low complexity domains with RNA, but I think I have run out of time. So what I will do is actually sort of leapfrog ahead and basically sort of provide you with an overall summary, which is that there are in fact decipherable rules that govern the driving forces for condensate formation. We think we can extract this using the quantitative deployment of a stickers and spacers formalism. This is kind of an exciting time for us. So I'll end by acknowledging the contributions of key players. So Jungmo, whose work I talked about quite extensively, for Kandar, a graduate student in the lab, and we've benefited immensely from a collaboration with the Alberti and Tony Hyman labs. The part that I skipped over involved a fantastic collaboration with Steve Boynems at Stanford, driven by Alex Holhouse, but I will stop there, and hopefully we have time for some questions. Over to you, Mike.
All right, thank you so much. So before we get to those questions, I'm just going to make a few quick announcements. The first is that if you would like to learn more about the functional implications of intercellular phase transitions, you can do so by reading this biochemistry article, of which Rohit is the author. And with that, I'm going to turn it over to Sarah to tell you a little bit about a special issue of biochemistry, as well as take your questions.
Great, thank you. If you want to learn more about the biology and biochemistry of phase separation, I encourage you to check out Biochemistry Volume 57, Issue 17, which is a 2018 special issue focused entirely on membraneless organelles. You'll find a link in the slide deck and also at the top of the biochemistry Twitter feed at Biochemistry ACS. Now, let's get the audience involved in the discussion. I want to start with a question from Tad, who asks, for sticker regions, is there a pattern to the residues that surround the tyrosines and arginines?
The answer is yes, and we're just starting to get to that issue in some great depth. So there are two things that have emerged in recent analyses. The sequence context of these stickers will certainly play a very important role in both governing the effective strength of a sticker, but also, so that would be the intrinsic strength of a sticker, but it will also contribute directly to how the stickers actually see one another, because the spacers and even the immediate context can modulate the sticker-sticker interaction. So there is definitely a conditional logic, if you will.
And then a related question from Edward. Do divalent metals such as magnesium cause these condensates to form at lower concentrations?
So the short answer is absolutely yes. So the phenomenon of phase separation is very much dependent on solution conditions like salt concentration. And so as a sort of, like I said, a coarse-grained theoretician, I sort of used the y-axis to depict inverse interaction strength, but that inverse interaction strength, of course, is controllable by various excipients, including salts, and the valence of the ions that one adds will definitely matter. Depending on the charge content, divalence will, either sort of help chelate around specific sites and drive phase separation more actively. So you can also imagine it's not just the valence of the solution ions, but also the extents of the post-translational modifications that add sort of additional charge and or change the valence of the charge that could contribute quite significantly to the driving forces for phase separation.
Elizabeth is asking, is there a way to predict which proteins will interact based on the sticker spacer patterns? Like, could you do a predictive analysis on a genome and sort of uncover more of these condensates?
So there are two ways to do this, right? So one is I become dangerously glib and tell you that what we've learned for FOS or HNRNPA1 or, actually something like 40 odd sequences can be transferable en masse to every protein in the proteome, and therefore I devise some clever machine learning approach and turn this into a server that predicts phase diagrams. That would certainly be lovely, but I think we are, as I responded to the first question, there are key issues that we're still trying to sort out, and that has to do with the effects of spacers, their sequence encoded effective solvation volumes, and the patterning of stickers. So I could certainly have, for a given amino acid composition or sticker composition, I could have a whole array of linear patterns of stickers that will contribute differently to the driving forces for phase separation versus actually precipitation, right? And so that's something that we've uncovered. And so before we turn something into a server, basically what we're trying to do is make sure that A, we have a handle on the rules, and the best way for us to gain a handle on the rules is through the types of simulations that the simulation engine Lussi will enable, and also another engine called PIMS that Alex Holhouse has developed, that when applied to an assortment of sequences will yield, hundreds upon thousands of sequence phase diagram relationships from which I think we can extract these rules. So I did punt on the answer. I recognize that, Elizabeth, but hopefully what you heard was that we are on it, and that the hope is that we will get to something that I think you desire within the next year or so.
He's asking, can you expand on what you mean when you mentioned something along the lines of the false notion that LCDs are what is crucial to phase separation?
Yes, so I think, well, maybe I spoke too heretically. So I think one can always be tempted to sort of look at a sequence and identify the low complexity domain and sort of pretty much assert a priori that it will be the driver of phase separation. That may well be true when one excises that LCD from the context of the protein. But when put in the context of the neighboring domains, it may well be that the interactions that are provided by the LCD in the context of that particular protein actually make it contribute more as a spacer rather than a sticker, for the simple reason being that a lot of the specific interactions or higher affinity interactions are coming from the folded domains. So let's say the protein RNA interaction domains. And instead of speaking in sort of vague terms, I can give you a very concrete example, and that would be the protein G3BP, which we've worked on together with Simone Alberti and Titus Franzmann, where it turns out that there is a dimerization domain. There are RNA binding domains which are folded. There are two middle intrinsically disordered regions which one intuitively would have, or at least as a knee-jerk response, have said, oh, those should be the players that drive phase separation, when in fact only one of those two actually sort of enables phase separation, and does so really as a regulatory region, whereby it sequesters and a third intrinsically disordered region, which is arginine rich, into sort of intramolecular interactions. And only when the concentration of unfolded RNA molecules is high enough are those sort of sequestered arginines released to enable phase separation. But then there's an additional intrinsically disordered region that fits the bill of being a low complexity domain, which contributes, as Paul Taylor has shown, directly to the fluidity of the condensates, right? So that would be a fairly striking example of the low complexity domains doing something that's very context dependent, because the major driver of phase separation in the context of G3BP are the heterotypic interactions involving G3BP and RNA, not the homotypic interactions that involve the intrinsically disordered region.
So I have a question from Editor-in-Chief Alana Shepard. She says, could you comment a little bit on the potential for non-natural backbones of these polymers as artificial linkers to control or drive or regulate phase separation?
Oh, absolutely. I'm so glad you asked that question, Alana, because yes, I think one of the major motivations for all of us is to try to understand sort of the biological role of these molecules that drive phase separation. But a perfectly orthogonal interest would be a chemical biology one, which is to actually design de novo condensates. And so long as one can actually incorporate unnatural amino acids, be they side chains, or let's say go from amides to esters, or amides to something else along the backbone, then absolutely one can sort of control phase separation in sort of a very, very interesting and clever way, whereby you can sort of pretty much decouple side chain and backbone interactions, and therefore achieve much more modularity in the control of the driving forces for condensate formation, which tends not to be the case in sort of biopolymers, where one has to always depend or sort of work out the particular context dependence. So absolutely, I think being able to, let's say, incorporate, for example, in the context of the work we've done with Mike, showing that the linkers actually directly control the cooperativity of phase transitions, and therefore the demarcation between whether you get a percolated droplet versus a test tube spanning network, and therefore essentially a gel versus a condensate that sort of can have gel-like properties. That design is essentially completely enabled by changing the properties of the linkers. In collaboration with Mike, we focused mainly on sort of naturally occurring variations, but there's absolutely no reason why we cannot start incorporating unnatural linkers or synthetic linkers. Absolutely. In fact, that would give a sort of more puritanical control, if you will, over the excluded volumes or effective solvation volumes.
So I have one final question about the physiological relevance of phase separation. And so, the cell is not an equilibrium system, and in a lot of cases of phase separation, it's things like mitosis or oogenesis. And I was wondering if you could talk a little bit about how these active physiological processes contribute to phase transition.
Absolutely. So I'll sort of try to be very quick here. So there's one feature of condensates inside cells that always comes across as being quite striking, and Frank Jülicher and Chu-Fan Lee sort of made this observation that condensates inside cells tend not to just grow willy-nilly to sort of fill out the cell. Instead, what you get is a sort of, there's an arresting in the sizes of the condensates, and so this looks like emulsification. And so one way you can sort of get emulsification is actually via active processes that Chu-Fan Lee and Frank have talked about. We've started to demonstrate that you can actually get this by certain sequence encoding features as well. But of course, the other attribute would be, and I'd say a major contribution perhaps of active processes, this is purely hypothesis, to condensates would be in the maintenance of the fluidity of condensates. So that way, the aging property that I touched upon might actually be ameliorated by the expenditure of energy in a non-equilibrium system.
Well, thank you, Rohit, for your presentation today. If there's one important lesson that our listeners should know, what would that be?
Stickers and spacers for all. I think there's a lot of deep interest in trying to figure out where structure plays a role. And maybe what we should start to think about is that everything I've talked about actually invokes structure, albeit not in the traditional sense, but more in the sort of structure that's encoded in the linear sequence that then gives rise to emergent properties on higher length scales, not necessarily on the length scale of just sort of specific structural elements. And so there is structure. We just need to widen our definition of what we refer to as structure. And I'd add a corollary message to that. This is an extremely important topic and exciting time. So hopefully I have persuaded you at least a bit to join the gang, so to speak.
Thank you for watching this presentation. ACS Webinars is provided as a service by the American Chemical Society as your professional source for live weekly discussions and presentations that connect you with subject matter experts and global thought leaders concerning today's relevant professional issues in the chemical sciences, management, and business.